So let me switch gears. I know that it's a very broad crowd. What I'm going to do is not give you too many technical details, but give you a feeling of where the field is going, where we want to go, and how AI could drive important biomedical research in many, many domains. But I'm going to focus more on diagnostics today, right? Let's get started. Aging is inevitable. All of us are aging. One can't stop that, right? Here is like a Wikipedia type of information. You know, when 50 years ago, the average life expectancy in the world was about, where is that? About 40s, in the 40 to 50 years old, right? Average life expectancy. But 2020, you ask how long are we staying? The time has increased. We live longer, right? The average life expectancy is about 71 across the globe. Some countries are more, like for example, some of the European countries have higher life expectancy, whereas India has about around 72. This is good that we're all living longer. It's nice to live longer. One of the unfortunate downside of that is that as we live longer, our cells also in the body age. As our cells in the body age, they stop dividing, right? They just can't divide any further in our body. But once they stop dividing, we were not exposed to all these kinds of diseases. Now we have a whole range of diseases in the body that sort of is linked to as we age, right? Because the cells stop dividing, and because of that, you have diseases of the brain that we are familiar with, diseases of the bones, cancer, metabolic disorders, what have you, right? So this is going to be a challenging task going forward for all societies, not just for Europe. For all societies now, we have to grapple with this. How do we understand as we age, how do cells become disease cells, right? Our understanding of that is very, very poor around the globe. Why is it that as we age, our cells become disease cells, right? It's very unclear. So there's a major unmet medical need. Can we develop good biomarkers for healthy aging? Because one good thing is that if we age healthy, then that's a good thing, right? Then we don't have to get to all these kinds of diseases. But that's a challenge, right? It's not clear how we will figure that out. So there's a big challenge. How do we detect these cells that go haywire as we age? Is there a possibility to detect? And if we can detect it well, can we then detect diseases early? As all of you know, as medical practitioners, you know more than I do, that if we detect diseases early, maybe the therapies could be very different. We still don't have proper therapies for many diseases still, right? And the therapy development will become more interesting if we try to understand how cells switch to disease cells and can we detect it, right? That's what I'm going to talk to you in a very broad sense today, how machine learning and AI could actually help us in that direction. The world over is a big revolution that's happening. I also teach at the ETH Zurich, I tell all the students, get on the computational bandwagon, whether you're a medical student, whether you're an engineering student, whether you're pure sciences, computation would be critical for you going forward, right? Understanding how there's new developments and new revolution that's happening in AI would be very critical, not only for medicine, but also for other areas, right? So let's take an example that all of you are familiar with. And let me walk you through This is something you know better, but just for my own understanding, I'll walk you through this. A lady gets diagnosed of a lump in the breast, right? So this is a mammogram. Somehow a disease is detected. Once you've detected it, what is the next step of things? To detect it, there are very nice imaging strategies. It could be MRI, it could be PET scan, it could be X-ray. There are many ways of detecting now, advanced methods to detect if there's some problem, right? Through a mammogram. Once it's detected, you take a piece of the tissue, and there is really state-of-the-art laboratory methods now to be able to analyze these samples. In fact, I went through the laboratory here, really very nicely established here at the hospital here. So there's a lot of histopathology, and now it's become digital pathology. There's a lot of digital pathology that one does, right? So once you have this, then all kinds of histopathology and genetic and biochemical analysis, you need to get to therapies. And even in therapies, there's really advanced therapies now, right? You have surgery, radiation, you could have immune therapy and so on, right? And all of this is really developing rapidly around the world, but yet we're way behind treating patients, right? Really correcting these kind of diseases is still hard, right? That's why all of you have a very big role. And I think this hospital for this ecosystem is so valuable going forward in the next phase. So early detection, of course, has a big advantage, as all of you know. I mean, this is the statistics already 10 years old, but it's worthwhile to go through the statistics. If you detected, let's say, prostate cancer at stage 1, the survival rate is very high, right? But if you detect it at stage 4, survival rates go down, right? Likewise for breast cancer, ovarian cancer, lung cancer, any types of cancer, but also any other kinds of diseases, not only cancer. If you detect early, the chances of survival is also, chances of therapies are also pretty good. But then there is still a big challenge. How do we detect as early as possible so that we can overcome this interface between healthy aging and disease, right? That's still a big challenge in the field, in the world. And that's what I'm going to address today. Is there a possibility to start to get there to detect? Not only us, there's a whole bunch of outstanding colleagues of ours around the world are pushing the frontiers for detecting early. So if you think of a disease, diseases don't happen overnight, right? Diseases takes a long time. And let's again look at, this is like a pathology before the 20th century. These are very classic drawings that Cahal made just by hand. Right? These are hand drawings of Cahal in the 1890s, where he could just by inspection, he started seeing that some cells become abnormal. He was observing it, right? Even today, we don't know how those cells became abnormal. The biology, the medical background of how these cells become abnormal, we don't understand, right? And that happens over a long time. For example, if you have a healthy tissue, slowly somehow few cells become abnormal. And these few cells that become abnormal start to generate a disease over a long period of time. So, for example, these are various types of cancer, and you see that it takes a long time. Cervical cancer, 6 to 10 years, breast cancer, those many years. For example, bladder, 20 years. So it's a long process where the disease starts to, show up in our body, right? So if these are so long, then it links to aging, because as we age, that's why we're becoming more susceptible to these kind of abnormalities and as they grow. So the idea is that can be pushed towards early detection. And there, pathologists have done a great job almost 50 years ago. Since 50 years, they started noticing that if you inspect cells in a tissue, cell morphology, shape, size, or nuclear morphology, everything changes in a disease. These are premature aging syndromes. You see that a nuclei of a single cell somehow has become broken down, right, as you see here. Or in Alzheimer's disease, you can see that there are changes in the cell and the nuclei, or in cancer, right, or in many other diseases. That means that a single cell that is sitting happily in a tissue, something changes, and the change then leads to alterations in their programs. And the program is that, as all of you know, we have trillions of cells in the body, but it's the same DNA. And if I take one of your cell and open out the DNA, it's literally 2 meters long, each one of a cell, right? Trillions of cells of the same DNA. But that has to be packed in very nice ways, so that to make a brain, you need to pack it differently, brain cells, or to make a bone, you need to pack it slightly differently. Or if you want to make a blood cell, you need to pack this DNA very differently, right? That means that the packing of this our DNA in our trillions of cells has a fundamental role in how it can integrate signals of all kinds, physical signals like mechanical signals and biochemical signals, and turn on many programs so that they stay healthy, right? And so we wondered that maybe, since the pathologists have been telling us that there are really subtle changes in the nuclear organization or the organization of the cell, we thought that maybe that's a good place to bring in the power of AI and ask, can we detect this? Can we use that as a biomarker, as a detector, right? So that's what I'm going to talk to you today. And when you think of AI, as all of you know, it's been a big revolution. This is one of the first papers. What people, and this won the last year Nobel Prize as well, one of the authors, what they realized is that in pictures, let's say I take a picture of all of you, there is a lot of information, right? By just taking pictures, I can detect who is male, who is female. Then by taking pictures, I can say who is young, who is old, because in the picture I can detect. Likewise, I can start stratifying the information, right? What they took a lot of pictures of human faces and realized that there's a lot of different types of data that you can group together. How the noses are set up, how the ears, and so on. That means that you can start learning information from those pictures. That's why, Tesla car works, because Tesla has learned where the traffic light is, where the stop sign is. Everything it has learned through these machine learning models. So likewise, in these kind of things, you can learn various aspects in those pictures, right? If you learn that data structure, then there's a very powerful way of saying, is a cell healthy? Is a cell aging? Is a cell abnormal? And so on and so forth. That's what I'm going to talk to you today in a very broad level, that looking at pictures like this, not of the faces, but of our tissues and of our cells and of how the DNA is packed in each of the cells, we can then learn data structures just like the way these data structures are learned. This is very generic. The technologies are now very advanced. It's really state of the art. It's rapidly growing. And what I'll introduce you today is some of the ideas that we can then implement to detect it. So instead of taking pictures of faces, what we do is take pictures of tissue biopsies or blood cells, lot of them, right? If we take a lot of pictures like this, then each of the picture, you can then peep into each of the cell. Inside the cell, as I said, you have this very nice DNA thread that's packed up in each of the cell, right? In each of the cell, there's a nice DNA thread that's packed up. Now, taking thousands and thousands of images of these cells and DNA, you can then use various types of learning models. You can use what is called supervised learning. That means that I say, tell me the size of the nuclei, tell me the size of the cell, tell me the size of some internal organelle in the cell, right? So I can learn by supervised learning, what is called. Otherwise, I can also learn through what is called deep learning, right? I just give data, I just learn whatever it is. This is what Tesla does, right? It uses really advanced learning models to just learn off everything that's there on the street, right? And you can then use those technologies to learn what is there inside the nucleus and cells. So what I'm going to show today is it's a very powerful biomarker of the cell's health and state of the cell's health, or is it disease. So when you want to learn, you learn many things. Like, if I take picture of this auditorium, I can learn many things. Who is young, who is male, who is female, who is wearing what kinds of dress, and so on, right? All that I can learn. Likewise, when you take pictures of cells, you can learn a lot of things. You can learn about how the nuclei and cell morphologies are set up. There's a number of parameters that you can learn. Or you can learn about how the DNA itself is packed. Many kinds of measures of how the DNA is packed. Which regions is more packed, which regions there's crowding, which regions is more empty, and so on and so forth. So what you've got is a whole lots of information on how the morphology, how the form of this cell in the tissue or in the blood is established, right? Now, as all of you know, form and function are very linked. So if I know the form, I can predict the function. Now, there's a whole new area which is called multi-domain data translation. This is not only in medicine, in biology, it's also in finance sector, it's also in traffic control. This is because data exists in different modalities, right? And in stock markets, everything, this is a very big area of how you take data from one type of one modality and transfer it to the other data modality. So what we have established in recent years is state-of-the-art methods. If you take a picture of a cell in a tissue, I can tell you what the profiles would be. What would be the protein expression profiles of the cell? What would be the metabolic profiles of the cell? By just looking at the picture, because I can learn what it can produce, what is the protein patterns, and so on and so forth. Right? So we have now used some of these methods, and I'll walk you through some examples of how powerful it can be. One simple example is I send all of our medical students and I show you pictures of different stages of cancer cells, right? And I'm going to tell this machine learning model to detect for me which stage of the cancer cell this is. Because that's hard. Just by looking, even with advanced training, it's still hard to say which cells are normal, which cells are different stages of the disease. So here is the model that because we have so much information of every cell and every nuclei, what we can do is, taking different cancer cell states, we can then group them. Which type of features represent a normal cell? Which type of features represent a stage 1 cancer cells or stage 4 cancer cells? So that's what we do, like different stages of the cancer cells. Just by looking at pictures, because this is very important, right? If we can bring this to the lab-based analysis, it would be tremendous, because on the flight, when the surgery is going on, I can actually check what's happening at which region. How do I know which is normal, which is abnormal? You will have a big advantage to be able to do that. And also, as I said, diseases don't happen overnight. It happens over a long period of time. It's very hard to trace a disease that progresses in our body, because how do I know? I have to come to the clinic every week to take an MRI image, and I have to do that for so many weeks and years to get an idea of how the disease is progressing. So now there are very nice methods of, if I have information on different stages of the disease, I can learn. For example, for me to go from Tumkur to Bangalore, if I just know some intermediate points, then I can learn how can I flow the traffic from Tumkur to Bangalore through this intermediates to optimize the path, the envelope of how the progression happens. In the same way, there are these transport models now that you can take pictures of your tissues or what have you and ask, as the disease progresses, if I had the 4 intermediate time points, I can then make the envelope of how the disease is progressing by learning how the changes is happening to the cellular states, right? I just took pictures of a disease. Let's say it took 20 years for the disease to progress. I took a picture at 5 years, 10 years, 15 years, and 20 years. I can then construct how the transport of cells from stage 1 to 4 progresses with these kind of advanced transport models, right? This is the other kind of structures, because then you can transport data structures in time. Then I can already know if somebody comes and with a very early stage cancer, I can actually predict what it would be later, what it would be in the past, right? And for therapies, this would be valuable, and I'll show you an example. So with these models, I'll just show you one example how it can be applied. This is a human breast tumor progression. In terms of tumors, breast tumors, one of the most places where the tumor is very aggressive is the ductal carcinoma that many of you might be familiar with. Is the tumors of the ducts, milk ducts, and somehow inside the ducts, cells become abnormal and they become, they rupture the ducts and become very invasive. There's a big medical challenge that I'll highlight now. And so what we do is take biopsies of various stages of the disease, starting from a normal tissue. These are tissue slices called microarrays. And we look at all stages of the disease. And each of the dot there is how the DNA is packed in each of the cells, right? And we score them now. We have set up a score for that, because the packing gives us a measure of if it is healthy, if it is different stage of the disease, and so on. And you can nicely see as the disease progresses, you see cells switching from a relatively normal blue color to red and red and red. Finally, it's highly metastatic, right? So looking at the way DNA packing is changing, what we can trace is the progression of the disease. So this is good, because now if we can actually trace the disease, what we want to figure out is how did it start in the first place? Can we transport how it changes, how the disease progresses with these models? So what we do is, again, use this deep learning model to learn all the cells that are there in the tissue. You just group all the cells and ask which of the cells belong to healthy state, which of the cells belong to disease state, and how did it move? How do these cells move from this kind of a healthy state to a disease state? And then in various pathology, various disease progression state, then we can actually ask what fractions of the healthy cells are starting to become disease cells. Right? Initially, most of the cells were healthy, and slowly it started becoming unhealthy and became disease. And from these kind of things, you can construct a pattern of progression. Because as I said, in the tissue, there is a whole different types of cells, right? There could be immune cells, stromal cells, cancer cells. There's all types of cells in the tissue. And now we can actually map how these tissue cells are reorganizing this in the tissue as the disease progresses. And what we found was a very striking thing, that even in the normal case, there are residual disease cells in the tissue. Right? There are residual disease cells in the normal state, and those residual disease cells starts to increase as the disease progresses. And the reason why I'm emphasizing this is that even today there's a medical challenge, which is going from a ductal carcinoma to invasive carcinoma is very hard to predict clinically around the world, not just in India, around the world. The challenge has been that after the lot of epidemiological studies in the last 30, 40 years, we now know that when a woman is diagnosed of a ductal carcinoma, only 30% of them become invasive. 70% of them don't become invasive. Although we treat all of them, it's an over-treatment medical problem that because we don't know how to distinguish at this stage, at the ductal carcinoma stage, whether this patient will become an aggressive metastatic tumor. Even today, it's clinically hard to predict. So what we do is we treat everybody, right? But what the studies is telling, a lot of rich studies around the globe, is only 30% become aggressive. So the question is, by looking at the stage, can one predict, can a medical student here predict which of the patients become aggressive, which don't become aggressive? That's a challenge to solve still, and we are not still solved, the field has not still solved. But taking kind of deep learning models, we are now beginning to have a handle on it, right? So the reason why it's important is, let's revisit the tissue. These are the ducts where it become rupture. The challenge is to say which one will rupture, which one will not. And as most of you know, tissues have what is called stiffness, right? The tissues are usually soft. These kind of, breast tissues, all kind of brain tissues are soft. But as the disease progresses, they become very stiff. Like, as tissues become stiff, they reinforce the cells to become more fibrotic. Tissue fibrosis increases. So the challenge has been that can we try and look at this and fingerprint using our method, whether learning the information from not one or two cells, we learn information from millions of cells now. We use about 3.5 million cells to learn which subpopulations of those cells are going to become invasive and which subpopulations may not become invasive, and we're still not there. We have a very good way of mapping them. I'm not going to go into the technical details here. And what we can actually see is that how these different types of cells, which ones are being more affected and which ones get less affected in terms of the way DNA is packed in that particular tissue. So the final goal is, by using these machine learning models, by looking at the ductal carcinoma, can we predict for that patient, is it going to be non-aggressive or is it become an aggressive tumor? So this is one type of work I wanted to introduce you by looking at it. I took cancer as an example, but you can take, we also do neurodegeneration. We get human postpartum slices from the UK brain bank of frontal dementia, Parkinson's, Alzheimer's, and learn how the DNA is packed in the brain tissue. Can we predict which regions become Alzheimer's-like, which regions become Parkinson's-like, which regions are dementia? And then we feed that back to the clinicians to see whether that would be useful. Of course, in neurodegeneration, the problem is, even if you detect, we don't have a drug to treat. That's the problem today, that we can know that people are decaying. I don't have a drug on the market to really treat neurodegenerative disease. But yet it will help the clinicians to sort of understand what could be the possible measures they can take if they can detect early Alzheimer's, early Parkinson's. So that's one part of the thing I want to tell you. But here, all I've told you so far is it requires that the patient already has a disease and I can take a tissue biopsy. But the challenge still is, can we detect even earlier? Because if we can detect earlier, then I have a big advantage, right? how do you detect early? If I look at the body, how do I know where a disease is? It's very hard to know which part, right? But that's why the liquid biopsies have becoming very popular, because if you take samples of urine, samples of blood, or any fluid sample, that has a lot of information of the whole body, right? That's why we go to the blood testing, yearly blood tests and so on, so that we want to be sure that things are, but they're still not that accurate, right? And any disease that grows in the body, whether it's neurodegeneration, cancers, diabetes, anything, they secrete signals, because they have to secrete signals to make the immune system not fight to reduce the disease. So they secrete signals. Eventually, these kind of signals land up in the bloodstream, right? Let's say I have a disease growing somewhere, it's secreted as signals, it's come to the bloodstream, the blood is circulating, so it's mixing with the blood. So in recent years, liquid biopsies has become a very big industry for early detection, because people try and catch DNA pieces from tissues, diseased tissues that are released, ctDNA as it's called, take out a little bit of blood, fish out whether there's disease-specific DNA in the blood. Then there are what are called exosomes. These are proteins that are released to the blood. People collect it and analyze it. They are now state-of-the-art, mass spectrometry methods, time-of-the-flight measurements to detect are there any disease signal in the blood coming from from different tissues. The other thing is some of the disease cells come to the blood. These are called circulating disease cells. For example, in tumor, it's called circulating tumor cells. They come to the blood. You fish out and collect, because this is 1 in billion or something. It's a very low concentration. People now have very advanced methods to collect the small number of circulating tumor cells in the blood and detect it, right? So what we thought was, let's try a completely different idea. As I said, DNA is packed. DNA is also packed in our immune cells. And what is the purpose of the immune cells? As all of you know, the immune cells are the first checkpoint, right? If I have an infection, immune cells have to fight the infection. If I have a wound, immune cells have to fight it. So immune cells in a body are the most sensitive cells in a body, right? So if there are signals coming in, our immune cells have to integrate, see that signal and react to the signal, right? So that means that bodies establish an immune response for any disease. So we thought that maybe that's the place to look for, because if we take a picture of all our blood cells, take pictures, then maybe we can actually say if the blood cells are seeing some disease signals in the body, or they're just being happy and healthy. Right? So that's what we do quite a bit now, major clinical trials ongoing. That idea is that let's take an example of a tumor. The tumor releases some signals. We collect the blood cells, it's a fairly easy way, take pictures of the blood cells, and use our deep learning models to differentiate. Is it coming from a healthy person, or is the cells that I'm seeing in the person, is it looking like a cancer patient? Is it looking like a neurodegenerative patient? Is it looking like a metabolic disorder patient? Right? By taking pictures of the immune cells, we can now stratify the health or the disease state of that human being. Right? These are human trials. So we now have major trials with the Swiss Red Cross to make a map of healthy people, to make a map of diseased people, to make a map of people who have other kinds of diseases. But also the other advantage is when all of you, since you are clinicians, you do therapy, it's very hard to know how the therapy has worked. Right? The only way we know if the therapy, for example, for cancer has worked is after the therapy, you go back and take a MRI or whatever kind of imaging, but the resolution is not that high, because you can't get a cellular resolution with these kind of even advanced scanning system. That means that some cells that are residual again relapse. That's why the relapse of a therapy is very high, because we are not able to know whether we have removed all the disease cells. So the idea is that can we use some of these kind of methods to trace therapies? After you have a therapy, can we just take the blood cell and ask, is a blood cell looking like a healthy cell? Because now if I remove the tumor by surgery or some therapy, then the blood doesn't have these disease signals anymore, right? So that means that our immune cells are no longer seeing the disease signal, and maybe I'm able to detect them. So I'm going to show you an example, or a couple of examples. One example is, if people come, healthy people and cancer people, can I detect it? Just taking pictures, right? Pictures of the blood cells. And what we now have is very high accuracy, greater than 90% now, by looking at the images of the blood cells in our body, derived from healthy donors, derived from cancer donors, we can detect with very high accuracy using this deep learning models. The other thing is, what we want to know is what type of cancer this person might have, right? And so what we do is, again, collect blood, take pictures, and we have tried with 3 types, and now we are trying with many other types of cancer, many other types of disease. We can then stratify that this group, this cohort has, let's say, meningioma, head and neck, or glioma cancers, right? Because the reason is head and neck cancers are very different than gliomas, because the type of signals they release are very different to the blood. And because they release different signals, the immune cells respond differently, and the DNA packing is very different. That's one example. The other is, if I go to a clinic for a therapy, can I trace the efficacy of a therapy, as I said, right? Whether the therapy has worked, whether the therapy has not worked. Again, we did a pilot study, and now our whole clinical trial is complete, and it will be available to all of you. What we decided to ask is, take healthy donors, make a map of how the healthy donor, let all of them sit, how the healthy donor blood samples look like, right? And then we make a systematic protocol, take blood cells before therapy, during therapy, and end of therapy, and ask if the therapy worked. So maybe now that our student colleagues have been listening to me for some time, let me ask them this question. What would happen to the blood cells if the therapy worked? What would you expect? Let's say you had in your hospital a person with some disease, and you had some way of measuring the disease using, there are many blood-based tests, and now you gave some, therapy for them. What would you expect? What would the immune cells do, depending on if your therapy worked? What would be your guess? You're all medical stars, no? One of you should. If I have, let's say, take cancer as an example, right? There's cancer that's grown in my body. Then I look at my immune cells. It is, inflamed because of cancer, right? So now I do a therapy. What will happen to the immune system if the therapy worked or if it didn't work? Who is your star student? I think you all know the answer, it's just you're all shy, right?
They should become normal or they should die?
So inflammation will go down if the therapy worked, right? Because the immune cells are highly inflamed because they have to fight with the disease. Now if the therapy worked, the immune cell inflammation will go down, right? If the inflammation goes down, then the immune cells will look more and more like healthy, right? Healthy and normal, right? That's what So that's what we trace. Here is healthy donors. This is before therapy. The immune cells are highly inflamed. Right? And then we start doing the therapy. We want to push the immune system back to its normal state. We want to trace that very well, right? So that's why the immune cells are such a very sensitive detectors of the physiology of the body that you can trace efficacy of therapies very well. And now we have a large trial that got completed, and what we now see is that, as all of you know, you take 100 people who have the same cancer, give them the same therapy, it doesn't work for all the people. It works only for some people. It doesn't work for many, same therapy, right? So now we can now stratify for which patient the therapy works, which patient the therapy doesn't work, and so on and so forth. I'm happy to talk to you if anybody is interested in such drugs. But the problem here is that this healthy, I want to compare whether my therapy has worked. I need a good reference map. I need a good reference to what is healthy, right? And you go to 10 doctors. We are very star doctors here. I go to 10 doctors. One of them tell me I'm not healthy. The other person tells me, no, you're completely healthy. The other person, so there's an interpretation which is subjective, right? Each person decides who is healthy and who is not. So when we collect healthy reference map, it's very hard to know because there's a very huge heterogeneity in defining who is healthy. Right? So now what we have started to do is a big public health project, which is what I was discussing with our colleagues here as well. And you could also help us in this public health project. The idea is to make I usually called it atlas, but our colleague here gave me a nice word, encyclopedia. Right? So what we want to now construct is an encyclopedia of our immune cells, how their DNA is packed in the immune cells, for healthy people. So we had a huge difficulty in Switzerland about collecting healthy donors, and what we decided is to partner with the Swiss Red Cross because they have a number of tests to collect blood and they implant it. All these blood banks, you can't take blood from deceased patients and put it on healthy patients. So we're now constructing a large number of donors, healthy donors of all age groups, male and female, collect blood, take pictures of their blood cells, use our deep learning models, and make an encyclopedia as you see here. Right? This is the encyclopedia, and we have many methods to analyze it. This is like the encyclopedia of being healthy, right? For 10,000 people with all the variability, each person depending on what they've eaten, whether they've had a cough, cold, things change. So that's why you need a huge statistics to define who is healthy. Then you get this map, right? Now, with this background gray map, now somebody comes to the clinic, right? You collect blood and ask, do you fall within this map? If you fall within the map, you belong to a healthy cohort, right? Like if I have this large statistics, any new person coming in to me, I can say whether you fall within the healthy or you're outside this healthy bracket. So this is what we are doing. We have been very successful. We have a very quantitative index to define human healthy aging now. Because as I said, as we age, there are changes that happens, but still you want to be in the healthy cohort. Then that's a very nice way. The societal burden to aging healthy is much better than aging unhealthily, right? So we made this map, and this is a Swiss map. It's a very homogeneous crowd. And what we want to do now is, given that we have this power of machine learning and AI to define who is healthy, now we want to develop a community screening program. Because then this could be very transformational, because we can then compare anybody around the globe to this map. Of course, the European map would be different, so we want to construct maps in Asia, maps in North America, and so on, and Africa. because the ethnic background defines our body and how we are treated and how we are healthy. Depends on whether I'm an African, I'm an Asian, I'm an European, right? This is a very European map, right, or what we have constructed now and what we are finalizing. So to do that, that's why I got excited when my colleague and friend, thanks to her who invited me, Professor Kamala, we were classmates in college, and when she invited me that you could come here, I got excited to tell all of you that we would like you as volunteers to our program. Right? So what we're now trying to do, or what we've done, is develop these methods in a very miniaturized manner. With a small drop of blood that you can take and imaging the blood cells, the DNA images of the blood cells. And because we have this reference map or the encyclopedia, then we can map them into either healthy, or we have a number of clinical trials. How did the DNA packing change when you had cancer, different types of cancer, as I showed you? When you have chronic stress, we have a map of how the DNA packing changes when you have chronic stress, metabolic disorders related chronic stress, and we have a map that we are building now when you have neurodegenerative programs that are set up in the body. How does the blood cell chromatin change, right? Then we can compare with these healthy donors, right? If we had this, then we have now miniaturized this so much that we want to bring it as a point of care solution, which means that you can sit in your home, take a small drop of blood, put it on your slide that we send, take some pictures using your mobile phone, and send us the pictures. Because we are building a global repository of pictures of blood cells across different ethnic background. Now we are building it for Switzerland, European cohort, but we want to build it in other cohorts as well. That we create this for Asia. India is a good example, or China is a good example, where we have a lot of people. And the idea is to create this kind of a imaging atlas or encyclopedia as a reference map for healthy aging, and as a reference map for who is going to deviate from healthy aging to a disease state, which direction that the individual is going. If we track it early, then the therapies are going to be very different in the next phase, right? And so there's a big push like this even for looking at pieces of DNA in the blood. There's what is called a single cell atlas program. That's also progressed very well in the last 5 to 7 years. But our idea was that since our immune cells are so sensitive, they react, we are making a map of the DNA of the immune cells so that we can take this to a community screening program and to figure out if we can convert this into a public health project around the globe. I mean, start with Switzerland, but around the globe. And try to bring it as close to early diagnostics that we can do. Right? It's we have a lot of philanthropic foundations who have gotten very excited about this program. They want to see if they can contribute. But what is lacking in expanding this to other countries like India is the governing rules of how patient data privacy is also important, right? And so we cannot send samples outside, we cannot send imaging data, any clinical data outside. So we have to figure out how to work with ecosystems like yours and others, where we can bring in these technologies to the local place. That is a very, as you know, our own maid in our house says that I have three mobiles. You send me this small thing, I take the pictures and send it to you. So it's very easily accessible. In a very remote place, it's easily accessible. What it needs is a powerful AI-based reference map that you can compare. And if you don't have that, what do you compare with, right? So that's what we are building. So this is what I wanted to tell you. I didn't want to take too much time. I'm already more than... So just let me just summarize why we think it's an important definition of how AI can play a role in modern medicine, modern clinical medicine. I started with the idea that cells in our body that Gehalt saw more than a century ago, he was able to see that things are changing, something is changing in the cells, and which is eventually going to become a disease tissue. And what I showed you is that inside the cell there is this DNA that's packed. By learning, using AI methods to learn this packing and how the packing changes as the disease progresses, as aging progresses, we have an exquisite map for not only healthy aging but disease diagnostics. And this, we think, has huge opportunities in resource poor context, because if I'm good access to a good diagnostic facility, I can of course try and see if I can be diagnosed. But even there, the technologies are changing. AI is revolutionizing, even the diagnostic digital pathology is revolutionizing. But what I showed you is cost-effective, mobile-based community screening that we can do by taking pictures on our mobile phones, bringing the power of AI to really see if we can make a early diagnostics possible in the next phase. It will take us some time, but our Swiss effort is getting published, getting really publicized quite a bit in all kinds of forums within the government there as well. And we would like to see if we can partner here in India. And when Kamala invited me, I thought Tumkur is a nice place as well, that we can bring this community screening to local people. That's what we want to do. We want to build a global map here. With that, let me thank the colleagues that I have. We have very talented colleagues that come from not only medicine, but engineering, computer science, biology, and a number of postdocs and a number of scientists. And the work that I do is in close collaboration with Professor Caroline Uller. She's a professor of computer science at MIT and Broad Institute of the MIT and Harvard. And our funding, we are very well funded through multiple sources for carrying out this work. Thank you so much for your interest. So if you have any questions, don't be shy, because your professors are there. Happy to answer, but if you have any other thoughts, write me an email. With this chat GPT world, if you just type my name, you know who this person is, and you can write me an email. And let's engage in a dialogue on how to bring not only these rules, but how to bring early detection schemes for diseases in this modern day world sooner than later, because we are all aging. And aging, as I said, is introducing a lot of diseases, and you guys who are getting trained now are the next beacons of solving this problem, right? We have a huge problem in the world today because it's aging, and all economies are investing heavily in trying to understand this aging, longevity programs. So you guys have a big future, but you have to learn computational techniques. Even though you are doing your medicine, that's going to be the central. So I also urge your colleagues who are defining the curricula to really integrate computation. That's going to revolutionize in the next 3 to 5 years, as you see, right? Just the fact that Tesla works should be an example that I can sit in a Tesla or in San Francisco, I can just order a car without a driver, it comes to your gate, right? That's already telling us how the revolution is happening in medicine as well, right? So, thank you so much.